Okay, so we're diving into the 2024 Nobel Prize in Medicine today. And this one goes to Victor Ambros and Gary Ruvkun. They uncovered this whole world of microRNA and basically changed how we think about genetics.
Yeah, it really is a paradigm shift. It's like they found this hidden language inside our cells, these tiny molecules controlling how our genes actually work.
So before we get into all that, let's start with the basics. We're talking about gene expression here, right? Like the central dogma of biology, DNA, mRNA, protein, that whole thing.
Exactly, the blueprint of life. DNA holds the instructions, gets transcribed into mRNA, which then serves as a template for building proteins.
Okay, so far so good. But then how does a skin cell and a brain cell end up so different if they both have the same set of instructions?
Well, that's the million-dollar question, isn't it? And the answer, or at least part of it, is gene regulation.
Gene regulation. So like how some genes get turned on in a skin cell and others get flipped on in a brain cell, even though they both have the same DNA.
You got it. It's like having a giant cookbook, but each cell only reads and follows the recipes it needs at a given time. And the really fascinating thing is that Ambros and Ruvkun discovered this whole other layer of regulation that no one even knew existed before. And it all started with a tiny worm.
A worm? Like what? Those earthworms I used to dig up in my backyard.
Not quite. This one's a bit smaller. It's called C. elegans. And it's been a rock star in genetics research for decades.
What makes this C. elegans so special?
Well, for starters, it's incredibly simple, which is actually a huge advantage for scientists. It's like a living, breathing blueprint for understanding more complex organisms, even us.
Okay, I'm intrigued. So how do these worms and their genes tie into this whole Nobel Prize-winning discovery?
Well, Ambros and Ruvkun were particularly interested in developmental timing.
Right.
How does a cell know when to grow and divide and specialize?
Yeah, timing is everything. Too fast or too slow, and things can go haywire.
Exactly. And they were studying these mutant worms that had some really strange developmental issues. One group, called Lin4 mutants, were developing incredibly slowly.
Like they were stuck in slow motion.
Precisely. And then there was another group, the Lin14 mutants, who were developing way too fast, almost like their biological clocks were completely off.
Okay, so you have the slowpokes and the speed demons, both controlled by these messed up genes. What did they find when they dug deeper?
Well, that's where things get really interesting. They were both working independently, focusing on their respective worm genes. And what they stumbled upon was something that would change the face of genetics.
Okay, now you've got me on the edge of my seat. Give me the details.
All right, so Ambros was studying this Lin4 gene, the one that, when mutated, made the worms develop super slowly. Now, back then, everyone thought genes mainly coded for proteins, right?
Yeah, that was kind of the whole point of genes, wasn't it? They were the instructions for building those workhorse molecules.
Right. So imagine their surprise when Ambros found that Lin4 wasn't coding for a protein at all. Instead, it was producing this teeny tiny RNA molecule. I mean, minuscule.
Oh, tiny RNA. But what was it doing if it wasn't making a protein?
That was the big question. Meanwhile, Ruvkun was busy unraveling the secrets of Lin14, the gene linked to the fast-developing worms. And what he found was that Lin4 wasn't directly controlling the production of the Lin14 protein.
So it wasn't like a simple on-off switch.
Not at all. It was like something was interfering with the Lin14 blueprint, preventing it from being read properly.
Okay, so we've got these two separate lines of research, both focused on these seemingly unrelated worm genes. How did everything finally click?
Well, that's where the real brilliance of their discovery comes in. They realized that the tiny RNA Ambros had found, the one from the Lin4 gene, it perfectly matched a specific section of the Lin14 gene that Ruvkun was studying.
Wait a minute, you're saying this tiny piece of RNA was acting like a homing beacon, targeting the Lin14 gene?
Precisely. They proposed that this tiny RNA, which they later named microRNA, was attaching itself to the Lin14 blueprint, effectively silencing it, like putting a do not read sign right on the instructions.
So instead of just turning genes on or off, you have this whole other layer of control with these little microRNAs fine-tuning how much of a protein actually gets made. That's incredible.
Right. It was a completely new way to think about how genes were being regulated, a paradigm shift. But keep in mind, this was 1993. The idea that a little piece of RNA could have this much power, well, it was almost unthinkable.
I bet a lot of eyebrows were raised when they announced their findings. After all, this was just in some worms, right? No way this was happening in humans. Or was it?
You're right to be skeptical. Most scientists thought it was an interesting finding, but nothing more. Little did they know this was just the tip of the iceberg.
So they found this whole new way of controlling genes, but the scientific world wasn't quite ready to rewrite the textbooks just yet, huh?
Well, you know how it goes. Extraordinary claims require extraordinary evidence. Finding this unique type of gene regulation in a worm was one thing, but to really turn heads, they needed to find it somewhere else.
And I'm guessing that's exactly what happened.
You know it. It took a few years, but in 2000, Ruvkun's team found another piece of the puzzle. They discovered a second microRNA gene, which they called LET7.
LET7, what's so special about that one?
Well, unlike LIN4, which seemed to be specific to worms, LET7 was found in a whole bunch of animals, even humans.
Whoa, hold on. You're saying they found a similar gene in creatures that are completely different. That's like finding a hidden message that's been preserved through millions of years of evolution.
Exactly. It was a huge clue that this wasn't just some weird worm thing. MicroRNAs had to be doing something fundamental to life itself. And the discovery of LET7 was like firing the starting gun for a whole new field of research.
I bet labs all over the world were suddenly scrambling to find more of these microRNAs.
Oh, absolutely. And it was like opening a treasure chest. It turns out that our genomes are jam-packed with microRNA genes.
Jam-packed. How many are we talking about here?
Well, we now know that humans have over 1,000 different microRNA genes, each one potentially fine-tuning the expression of hundreds of different genes.
Wow, that's mind-blowing. It's like discovering a whole new control panel inside our cells with thousands of switches and dials we never even knew existed. But how do these microRNAs actually work? I mean, how does this tiny piece of RNA actually silence a gene?
Well, it's actually a pretty elegant process.
Yeah.
It all starts in the cell's nucleus. The microRNA gene gets copied into this longer RNA strand, kind of like a rough draft.
Okay, so you've got this draft, this long RNA molecule. What happens next?
Well, this is where things get really interesting. There's this special protein called dicer.
Dicer.
Yeah, dicer. It acts like a molecular editor, trimming and shaping that long microRNA into the perfect size and shape to do its job.
It's like getting fitted for a tailor-made suit, but for silencing genes. Okay, so now we've got our perfectly tailored microRNA. What happens next?
It moves out of the nucleus and into the cytoplasm, which is where all the mRNAs are hanging out.
And the mRNAs, they're like the blueprints for proteins, right?
Exactly. And here's the amazing part. Each microRNA has this unique sequence, kind of like a homing beacon, that guides it to a specific set of mRNAs.
So each microRNA has its own target, its own mRNA soulmate it's searching for in the cytoplasm.
That's one way to put it. And when the microRNA finds its target mRNA, it binds to it.
And that's when the silencing happens.
Exactly. It can happen in a couple of ways. Sometimes the microRNA just blocks the mRNA from being translated into a protein, essentially hitting the pause button on protein production.
So it's like setting up a roadblock right before the protein factory.
You got it. No mRNA allowed. But sometimes it gets even wilder. The microRNA can actually trigger the destruction of the mRNA altogether.
Whoa. Talk about taking out the competition. It's like the microRNA is saying, nope, not even going to let you get to the factory. You're done. But hold on a second. If a single microRNA can target multiple mRNAs, and one gene can be regulated by many different microRNAs, doesn't that get incredibly complicated?
It does. It creates this mind-bogglingly intricate web of gene regulation, which is precisely why this discovery was such a big deal. It's not just about a new molecule. It completely changed how we think about how our genes are controlled. It really was a paradigm shift for the field of genetics.
It's like realizing that the instructions in that cellular cookbook we were talking about, they aren't just recipes. They're part of this incredibly complex culinary language with its own grammar and secret codes. But I have to ask, with great power comes great responsibility, right? What happens when these micromanagers get out of whack? So we've got these microRNAs, these tiny puppet masters pulling the strings of gene expression. But what happens when, those strings get tangled? Do the puppets start improvising a bit too much?
It's a great analogy. And you're exactly right. When microRNA regulation goes off the rails, things can definitely go haywire. Think of it like a symphony orchestra. You need everything perfectly timed, everything coordinated for the music to sound right.
But if the conductor, our microRNA in this case, starts, maybe taking a few too many liberties with the tempo.
Exactly. Instead of a beautiful symphony, you get total chaos. And in the case of our cells, this chaos can manifest as disease.
So we're talking about real-world consequences here. Do scientists have any, concrete examples of how messed up microRNAs actually cause diseases?
Absolutely. One example that really highlights this is a rare disorder called Dicer-1 syndrome. Remember the dicer protein we talked about earlier? The one that trims and shapes the microRNA?
Yeah, the microRNA tailor, making sure everything fits just right.
Right. Well, if the gene that codes for that dicer protein gets mutated, it can lead to dicer 1 syndrome. And people with this syndrome have a significantly higher risk of developing certain types of cancers.
So if the tailor isn't working properly, the microRNAs don't get their perfect fit, and things start to, well, unravel.
Exactly. It really highlights how crucial that one protein, and by extension, properly functioning microRNAs are for keeping our cells running smoothly. And really, we're just beginning to scratch the surface of microRNA involvement in human health.
It's almost like microRNAs are this hidden network influencing a whole bunch of different processes in our bodies. What other areas are scientists looking at where microRNAs might be, causing a ruckus?
Well, researchers have found links between abnormal microRNA activity and a whole range of diseases. We're talking heart disease, diabetes, even neurological disorders like Alzheimer's disease. They're even involved in problems with development before birth.
Wow, that's a lot to take in. This tiny molecule is involved in so many different aspects of our health. It makes you wonder, if scientists could figure out how to, adjust these microRNAs, could we actually fix things? Could we treat these diseases?
Now you're thinking like a true innovator. And that's one of the most exciting frontiers of this research. It's not just about understanding what goes wrong, but about developing entirely new ways to treat diseases.
So instead of, say, taking a drug that affects the entire body, you could have a microRNA-based therapy that targets a very specific problem with, pinpoint accuracy.
Exactly. It would be like using a scalpel instead of a sledgehammer. And the potential is huge. We could potentially detect diseases much earlier by spotting tiny changes in microRNA levels, or even develop treatments that target specific microRNAs to correct those cellular malfunctions we've been talking about.
It's like something straight out of a science fiction novel. Are there any, promising leads on how to actually make these microRNA therapies a reality?
Oh, absolutely. There are already clinical trials underway testing microRNA-based treatments for various cancers, hepatitis C, even some genetic disorders. It's still early days, but the initial results are looking very promising.
Wow, it's incredible to think that this tiny molecule, this little piece of RNA, could hold the key to treating so many diseases. It really highlights the power of basic scientific research, doesn't it? I mean, who would have thought that studying a 1 millimeter long worm could unlock such profound insights into human health?
It really goes to show sometimes the biggest discoveries come from the smallest places. And to think, there are probably countless other microRNAs, other tiny mysteries still out there waiting to be discovered.
Well said. And on that note, we come to the end of our deep dive into the amazing world of microRNA. We journeyed from the depths of cellular processes to the forefront of medical research, all thanks to these tiny molecules and the brilliant minds who've worked tirelessly to unravel their secrets. Until next time, stay curious.